Hello everyone. Today we are going to continue the chapter Biotechnology and its application, and in which we are going to cover the NCERT session 10.2.2 and 10.2.3. In this, we are going to see more about gene therapy and molecular diagnosis. So, let's go ahead. Disclaimer. What are going to be today's learning objectives? First, we will get to define what is gene therapy is. We have talked about this gene therapy word a lot of time in previous chapter, but today we are going to learn it by the help of an example. We will also see a historical context of gene therapy, in which we will see how it has been used to cure a 4-year-old girl who was suffering from a genetic disorder. Then we will identify the limitations of conventional diagnostic methods, and then we will enlist and briefly explain the principle of key molecular diagnostic techniques. So first we will talk about what are the limitations which we face in this conventional diagnostic method, and then later we will explain the principle of key molecular diagnostic techniques, and we will see a few examples of it also. So yeah, let's go ahead. So today's index will be like this. First we will cover gene therapy, which is 10.2.2 session of NCERT. Then we will see molecular diagnosis, which is 10.2.3 session of NCERT. And then there will be a gist of all those slides which we are gonna study in today's class, all those topics which we are gonna cover in today's class. There will be a summary slide. Then there will be some NCERT based questions which we're gonna solve together. Then there will be some keynotes for you guys. And lastly, there will be some interesting questions that you guys gonna solve yourself. So gene therapy. What is gene therapy? So according to the definition which is given in NCERT, and if with the help of this slide you can also answer NCERT exercise question number 8. So gene therapy is a collection of methods which allows for the correction of a gene defect that has been diagnosed in a child or embryo. It involves inserting a normal functional gene to compensate for the non-functional one. So what does it means? So it's saying that gene therapy is a collection of methods in which what this is doing. So it is correcting a defective gene, a gene which have some mutation, which have some problems. So it is going to correct that, and how we have identified that this gene is not fine and this gene have some abnormalities, we can diagnose it in the child or embryo stage, and then what we can do, we can insert a normal gene. So that gene which is causing that disease, it is defective, it has some defect, it has some mutations, anything, but there are different types of mutations, but we are not going to talk about that in this class. So it might have some mutations, some insertion, deletion, anything. So how we can treat it? So we can insert a normal gene instead of that defective gene, and what it's going to do, it is going to, that now gene is going to compensate for that non-functional gene and going to work properly in that person's body as a functional gene. So you will get to understand it more when we will talk about the examples. So first let's see this picture, what this picture is depicting. So here has been showing that there has been a cell with non-functional gene. So this gene and this cell has a non-functional gene. What we did, we added the DNA containing a functional version of the lost gene. So this gene as shown in this cell, it is not working, has some defect. What happened in the next one? We have added a functional DNA with that functional version of that lost gene, and now this cell is functioning normally. This cross is defining that this cell is not working properly, But when we added a DNA with that proper gene, with that gene which is functional, that cell now became automatically functional and it is behaving normally. Second picture is also depicting the same thing, but it is, I think, more quite clear that this was a cell with non-functioning gene. Means this gene is present in the cell, but it has some abnormality, it has some defect, that's why it is not functioning well. But when we have added a DNA which contain the functional version of this lost gene, then what happens? The cells start functioning normally. So this is what actually gene therapy means, that we can insert a normal gene, a functional gene, to compensate the non-functional one, which we can diagnose at the stage of child or embryo stage. Okay. So, clinical application of gene therapy. So, first time when we have used this gene therapy clinically, first time when we have used this on a human. So, it happened in 1990. Who was a patient? The patient was a 4-year-old girl. And what was the disorder? What that girl is suffering from? So, she has this adenosine deaminase deficiency. So, what actually happens if you have this deficiency? Your immune system is not going to mature and your immune system is not going to function properly. So, and what was the cause? Why this happened? Why this disorder happened? So, this deficiency happened because the gene which encode for this protein, okay, which encode for this enzyme, that is adenosine deaminase, that gene has been deleted in that girl. It is not present in that girl. So, this is what it looks like. Adenosine deaminase, this enzyme which is coded by the gene, that gene is present on chromosome number 20, but in a patient who is suffering from this adenosine deaminase deficiency has this mutation, that this gene has been deleted from that patient. Okay, so this is the pictorial representation how there has been the deletion of that gene, particularly which is encoding for adenosine deaminase enzyme. So now, what is the consequences? We know that this girl is a 4-year-old girl who has this adenosine deaminase deficiency, enzyme deficiency. Why? Because there is a deletion of that particular gene from the chromosome number 20. And what was the consequence of that? Is that this her immune system is not functioning properly, because this enzyme is crucial for the proper functioning of immune system. Its absence lead to SCID. And what is SCID? SCID is a disease which we call severe combined immunodeficiency. So what happened? So because of this ADA enzyme is not present in that girl, she is suffering, her immune system is not working properly, and she is suffering from SCID, that is severe combined immunodeficiency. It is a very rare and inherited genetic disorder. Okay, this has been inherited, that prevents the immune system from maturing, leaving children born with little to no immune defense. This girl, like, if she's even, what happens, so even normal person, if they get cold, their immune system work properly, but their immune system is so weak that they don't have any immune defense, that even a normal cold can become a very big problem for that girl. So she is suffering from that. Now so now we know that because of this ADA, this adenosine deaminase gene deletion, she is suffering from all those things. So now what are the different approaches so that we can treat this ADA deficiency? First is bone marrow plantation. And what is the process of bone marrow transplantation? We replace the patient's defective bone marrow with the healthy marrow from a compatible donor. So what we do in this bone marrow transplantation, we replace this patient's defective bone marrow with a healthy bone marrow. Okay, but that donor has to be compatible. And what actually the outcome is? Potentially it cures the disease, but finding a suitable donor is very difficult. You can't find a suitable donor in this bone marrow transplantation thing. Mostly body rejects others bone marrow transplantation. So it becomes very difficult to find a compatible donor. So this is the first approach to which we can treat this ADA deficiency, that we can replace the patient's defective bone marrow with the healthy bone marrow. So now the whatever the cells will, the new cells will be produced, it is going to have our this gene, our ADA gene. But as I said, due to finding a compatible donor, it is very difficult to find. So now what is the next approach? First we talk about bone marrow transplantation. Second is enzyme replacement therapy. In this we try to replace the enzyme. So what is the process of this therapy? So first, the patient is given functional ADA through regular injection. So what in this therapy doctors do? They give patient the ADA enzyme which is actually working, which is functional, which is producing this adenosine deaminase through injection. But you have to give that injection regularly. Okay, because cell is not producing this ADA. You are giving it through injection. So you have to give it regularly on regular intervals. Outcome, what is the outcome? It is not a permanent cure. Okay, it is not permanent cure because cell is not producing it. You are giving it through regular injection, and it requires lifelong period. Means bone marrow is not producing in this bone marrow transplantation. Bone marrow is going to produce those cells which are it's going to have that enzyme. But in this, we are giving it from outside, so it is not a permanent cure. It requires lifelong periodic treatment. So here is a pictorial representation has shown of enzyme replacement therapy, in which enzyme replacement therapy weekly injection with PEG ADA. So weekly that person has to give this enzyme. Okay. Now, first we have talk about bone marrow transplantation. Second, we have talk about enzyme replacement, in which we are what we are doing. We are replacing the bone marrow, which is now going to produce the cell with that gene. In second, what we are doing, we are giving the enzyme, but we have to give it to the regular interval because the body itself is not producing that enzyme. So cells are not producing those enzymes. So what? So we are giving it from outside, and we have to now give it on the regular basis. So now the third and most important approaches which came into light is the gene therapy with lymphocyte. So this from this session you can answer NCERT exercise question number 8, and now we will see that how this gene therapy can done with the lymphocytes. So what was the process? First we have to extract the cell. What is lymphocytes? So lymphocyte is a type of white blood cells. Okay, are extract. So this cell lymphocytes are extracted from the patient's blood. Then they are grown in culture outside the body. So now this is a pictorial representation of ex vivo gene therapy. Ex vivo here represent that experiment that has been conducted outside the living thing. Okay, ex means like the name itself suggests that the ex vivo means outside the living being, living body. So first what we have did, we have taken the lymphocytes from the patient's blood, and now we have grown it in a culture outside the body. Of that picture, I'm gonna explain at the last. So, after extracting that lymphocytes from the human body and culturing it on, culturing it, what we do, we will add a functional ADA, that is adenosine deaminase cDNA, that is complete DNA, into that lymphocytes. And insertion, how it was used? So insertion, how was it was done? So insertion we do using a retrovirus as a vector. So when we studying this biotechnology principle and processes, we have talked about the some vectors, in which we have talked about retrovirus also, that we can use retrovirus as a vector in human body, de-armed retrovirus. Because what this retrovirus used to do, that retrovirus has the ability to convert the pro-oncogene into oncogene. Okay, means it can trigger cancer, can trigger tumor. But if we disarmed it, means then what it can do after disarming him, it can use as a vector. So insertion was done using retrovirus as a vector. Here we are seeing that how whatever we have studied in biotechnology principle and processes, we are seeing its application, that how it can be used actually. So now what happened? So we have taken lymphocyte, we have from patient body, now we have cultured it outside in the optimum condition and all. We have cultured that cells, lymphocytes. Now we have inserted a ADA, adenosine deaminase complementary DNA, into those lymphocytes using a vector which is retrovirus vector. So now what happened? So because we have inserted that ADA cDNA into that lymphocytes, now they are genetically engineered lymphocytes which we are going to grow in a culture. After going it now, so now what happened? We have grown a lymphocytes which have corrected gene in it. Okay. Now because it has corrected gene in it, so now we are going to return it to the patient. We are going to reinfuse the corrected cells to the patient. So now I will explain this diagram. So what this diagram says? So this is a ex vivo gene therapy. So first what happened? First we have removed the ADA deficient T cell from the SCID patients. Okay. Now we have cultured that T cells. Here you can see that, okay, take the example of lymphocytes. We have taken the lymphocytes from the patient, okay, SCID patient. We have cultured that lymphocyte. Now here what we did, here we have take bacterium which is carrying DNA vector with clone normal ADA gene. Okay, so now this is a bacterium which is carrying DNA vector with clone normal ADA gene, a ADA gene which is functioning properly. And now what is it is? It is a genetically disabled retrovirus. It is a vector. Now we will combine this, we will clone this ADA gene, okay, into this virus. And now this virus infect the cells with a retrovirus that contains the normal ADA gene. Now we will now we will infect this, you know, lymphocytes which have already been this inserted. We have inserted our normal ADA gene using this retrovirus. Now we will culture it, and now what we will do, we will reinfuse the ADA gene back into the SCID patient genetically alter lymphocytes, which is now going to produce the ADA gene. Okay. So what happens in this gene therapy? So first what we will do, we will take the lymphocytes from the patient. What are the lymphocytes? Lymphocytes are the broader category of white blood cells. So we took that lymphocytes from the patient. Now we know, now we culture that lymphocytes, and here it is T cell because T cell is one of the, like, lymphocytes are categorized into T cell, B cell, and natural killer cells. So here is, that's why it has taken one example of T cell, but I will speak lymphocytes only. Okay. So now they have taken lymphocytes from the patient who is suffering from this SCID, that is severe acute immunodeficiency. So they have taken this lymphocytes from the patient. Now they are culturing that lymphocytes in laboratory. Okay. Parallelly, what they did, they take our they did what we said, studied in this last chapter. They use this DNA recombinant technology. What they use, they have taken this they have taken the bacterium which is carrying this normal ADA gene, normal gene which is producing this adenosine deaminase enzyme. And now what they did, and took the other vector which is took this vector which is what, which is a retrovirus. So now this retrovirus, now who has this functioning ADA gene, this retrovirus, we are which is going to act as a vector, we are going to infect that lymphocytes with this retrovirus. Who, what this retrovirus is containing, it is containing the normal ADA gene, a gene which is going to produce a normal functioning adenosine deaminase. Okay, so now because we have infected that lymphocytes also with this retrovirus, now this lymphocytes are have transformed, and now it's functioning properly, that it is producing this ADA enzyme. Okay, now we will take this lymphocytes and now we will reinfuse this ADA gene, okay, back into the SCID patient. And now what it has, it has genetically altered lymphocytes which is going to produce the enzyme. So this is how it works. First we have taken the lymphocytes from the patient only, then we have cultured their lymphocytes, and parallelly what we did, we used this recombinant DNA technology where we have taken our gene of interest, our ADA gene, we use vector as retrovirus. Then what we did, this retrovirus has took that ADH functional gene to this. We have infected that retrovirus with this functional ADA gene to that lymphocytes, and now this lymphocytes has transformed. Now it is producing ADA gene. So now this transformed, now this ADA gene producing lymphocytes have reinfused into the patient, and now patient is able to produce the ADA enzyme. Okay, and now this patient is what, it is having a genetically altered lymphocytes which is producing ADA enzyme. So this is whole gene therapy. So what is the now outcome? So the outcome here will be that now, okay, that patient is going to have this genetically altered lymphocytes, but here we have to understand that this is not a permanent cure. Okay, why this is not a permanent cure? Because lymphocytes are mortal cells, means that it is going to die. So that the person is producing ADA enzyme only then until that lymphocytes are living. Once that genetically modified lymphocytes die, then this again there will be no production of this ADA enzyme. So that's why this approach is not a permanent cure. So the patient requires periodic infusion of these corrected cells. In patient we have to infuse this genetically modified lymphocytes in a particular intervals and again. This is not like again and again like what we are doing it in enzyme therapy. It will be work more than that, but we have to do that. So this is not a permanent cure. So now what is the permanent cure via gene therapy? How we can do gene therapy so that this become the permanent cure? So the ideal solution here is that for a permanent cure, gene therapy must be performed at an early developmental stage. At this gene therapy, all those things which we are doing, we should do it at a very early development stage. What was the proposed method? we can do what? We can isolate the functional ADA gene. We will introduce this gene into stem cells, which is isolated from the patient's bone marrow. We know that what is stem cells? Stem cells are the cells which has the ability to differentiate into any kind of cells. So what they are saying, we will isolate a functional ADA gene, then we will introduce this gene into the stem cell which they have taken from the patient's bone marrow. This must be done at an early embryonic stage. So this we have to do when that child is in the embryo form only. Okay. So what is the reason that doing this will become a permanent cure? Because bone marrow stem cell can become all blood cells and renew themselves. We know that that this bone marrow do what? They can become all type of they produce these blood cells and they can even renew themselves. So if we are doing this using the stem cell for bone marrow, then they all can become blood cells and then they will renew themselves only. We don't have to give it on a particular intervals. So using them ensures a lifelong supply of ADA producing cells for a permanent cure. What this will ensure? This will ensure now this patient whose this stem cells, we have used his bone marrow stem cell for this gene therapy and given them a perfect functional ADA gene. So this will ensure them a lifelong supply of this ADA producing cells and this will ensure a permanent cure. Now, so till now we have talked about what gene therapy. We have taken one example how first we have clinically used this gene therapy in a patient of with deficiency of this ADA enzyme. Now, we will move to molecular diagnosis. So, before going to this molecular diagnosis, there are some limitation of conventional methods. So, what are those limitations? So, the limitations are effective treatment of a disease needs early diagnosis and detection. So, if you want to treat a disease effectively, if you want that person should be disease free, we have to identify that disease, diagnosed as disease very early. What happened in cancer patients? We have seen examples that it's not like that cancer is, I know that cancer is not that curable, but what happened in most of the case? So people got to know about their cancer in stage 3 and stage 4 because the diagnosis is very late. So, if we were able to diagnose early, detect early that disease, it will become very easy for us to treat that disease. Traditional method of diagnosis, which includes serum analysis, urine analysis, are often not effective for early detection. So, what happens? So, when the pathogen goes inside a body, so it's not like that in a moment it will start dividing so fastly, and we can see a lot of there will be like a lot of symptoms. No, it won't be like that. Even in this serum analysis, urine analysis, they are not like in a very early stage, we are not even able to detect those pathogens and all because they are in very small quantity. With time, they are going to multiply. So, when they are going to multiply and when we are going to detect it using those methods, it will become very difficult to treat that disease. So, by the time a disease is detected by this method, the concentration of that pathogen or the physiological symptoms are already high. As I stated, the time we got to know the okay, this person is suffering from this disease, the concentration of that pathogen was so high, the symptoms became so severe that it became very difficult to get treated. And even if it's get treated, it what happens? This make patient very suffers more. So now, what should be the solution of that? This is the limitation. So here we can use this molecular diagnosis, which we're gonna talk up in upcoming slides. And what are the advantage of this molecular diagnosis? They allow for very early detection. They are very sensitive and specific. So, what is as we are detecting that disease at a molecular level, as the name suggests, molecular diagnostic, it allow us to detect it very early in a very early stage. And they are very sensitive and specific. It's not like that they are going to give us some false report. No, if they are saying that this pathogen is present in this human, it means it too present in that. So it is very sensitive and specific. So what are the example of this molecular diagnosis? Recombinant DNA technology, polymerase chain reaction, and enzyme linked immunosorbent assay, which is called ELISA. First two we have already studied, and we're going to talk in coming slides also. Third we will study for first time, I think. First molecular diagnosis is polymerase chain reaction. So we know, we studied in the last chapter that what actually polymerase chain reaction do. It amplifies the nucleic acid. So if there is a patient in which there is a pathogen, and even if that pathogen is in very small amount, okay, this polymerase can amplify that pathogen's nucleic acid, and we will get the idea, and we are able to see, okay, this person is like infected with this pathogen, and it's gonna amplify in upcoming days, so we can treat it beforehand so that person don't have to suffer a lot, and the concentration of pathogen remains limited, and the symptoms doesn't got so worsen. Okay, so what is the principle which we used here? That amplification of the nucleic acid, as I said, that there will be the amplification of the nucleic acid, that is DNA or RNA of the pathogen. A target DNA segment can be copied billions of times in a short period. So what are the application of these? One of the major application is that we can detect HIV in suspected AIDS patient. If we are thinking, okay, this patient might have AIDS, okay, but it is not showing right now so rigorously because HIV has not spread that much. So we can detect this HIV virus through PCR. It detects the viral RNA and DNA at a very early stage of infection when antibody test may be negative. Even if the antibody test is saying ki, okay, this is negative, this person is not AIDS patient, it is not suffering from AIDS. But what happened? Even if it has a very small amount of viral RNA and DNA of HIV, it can be detected, because what it will do, it will amplify that viral RNA and DNA. Detecting gene mutations. In cancer patient, mutations in gene are detected. It is also widely used in many other genetic disorder. So we can even use this PCR, okay, in detecting the gene mutations. And this gene mutations we can detect, we can, help, this PCR can help us to detect this gene mutations in many of the cancer patients, because we know that cancer patients do have a lot of mutation in their genes. So PCR can help us to identify that, and even it is help in identifying this gene mutation and all those things in many genetic disorders. Then there is something what we call RNA and DNA probing. So what tool it has been used? A probe. A probe, I think we have already studied this, but let's study it again. What is a probe? It is a single stranded DNA or RNA molecule which is tagged with what? A radioactive molecule, and this can bind to its complementary DNA or RNA sequence. So what is like principle of this method, this DNA and RNA probing? That this probe, which is a single stranded DNA or RNA molecule and which is tagged with a radioactive molecule, it will go and combine to its complementary or hybridize to bind to its complementary DNA in a clone of cells. So, how it is used in detection? So, the sample is then subjected to autoradiography. Okay, so here first you have to understand like how we can detect using this DNA and RNA probing. So, we are thinking that this a patient is suffering from some disease, and this disease is causing by the particular pathogen or whatever. We know a DNA sequence of that pathogen. कि, okay, this like DNA and this RNA might be like this sequence might be the if it is present, so we can say that this is present. So now what will happen? We will take a probe, we will make a probe which is a single stranded RNA or DNA, it depends on what we are detecting, and it is tagged with a radioactive molecule. We know that if there is a presence of that particular DNA or RNA in that sample, so it will now what probe will do? The probe will go and bind to that complementary region. Once it is bind to that complementary regions, what we can do? We can take that sample and then we can use this autoradiography, and when we can see, okay, there is a presence of this particular pathogen. So this is how we can use this DNA and RNA probing for the detection of diseases, diagnosis and detection of diseases. So how we analyze it? If a probe binds, it indicates the presence of gene of interest. As I said, if it go and bind to that particular DNA sequence or RNA sequence, we will say, okay, so there is a presence of the gene of interest which we were thinking that is going to be in that patient sample. The radioactivity will show up on the photographic film. So the radioactivity we are able to just see on the photographic film. What is application? So this technique is used to identify specific gene mutations, and gene mutations might be associated with disease like cancer. So this technique is widely used for identifying these specific gene mutations, because as I told, they can make this probe and DNA particular to their DNA sequences or gene of interest which they wanted. So this technique can be used for identifying the specific gene mutations, and that gene mutations can might be even associated with diseases like cancer. Next method is ELISA, that is enzyme linked immunosorbent assay. So what does this enzyme linked immunosorbent assay is about? It is all about antigen and antibody interaction. So we will see like how it works. So let's see. So what its principle says, that it's based on the antigen and antibody interactions. Now, how we detect the pathogen using this? What its application is? So, the infection by pathogen can be detected by the presence of either antigens or antibodies. Antigens include what? It includes protein or glycoprotein, and this is produced by whom? It is produced by pathogen. Whereas antibody is present by that human in the response of those antigens. So here I'll explain this whole figure. Before that, you will need to like cover the some few basic concept also. So here you guys know and got to know that this enzyme linked immunosorbent assay, which is commonly called and knownly called ELISA, it's based on this antigen and antibody interactions, and we can detect this infection by pathogen by the presence of either antigen or antibodies. We have different kind of ELISA, but here you just guys need to understand normal like a concept of ELISA, like how it is being used. So here antigen is what? Antigen is something which is produced by pathogen, and it is like mostly proteins or glycoproteins in nature. Alternately, it can be detected by identifying the antibodies also. You will understand it how it will detect it. These antibodies are synthesized by the host immune system against the pathogen. As I said, these are antibodies are what? These are produced by host immune system only against this pathogen. So now see how it works. So this is a microplate. Okay, you understand it is a microplate, and here we have this capture antibody. Okay, and we know that this antibody is specific to a antigen. So now what happened? We have this capture antibody. Okay, this capture antibody, what will happen now? The target antigen will come and bind to it. Once the target antigen will come and bind to it, what happens? So now the other antibody. Now which antibody? Antibody which have enzyme label, means it has a enzyme. What does this enzyme do? This enzyme has the ability to convert a substrate, chromogenic substrate. Okay, so now this has the ability to convert this chromogenic substrate into a particular color. So how now we will understand ki okay, this antigen or antibody is present in there. First we have this capture antibody, which we know that is specific against this target antigen. We here the target is antigen. Now what we will do, because this antigen which is specific against this antibody comes and bind to this antibody. So after this, now how we will understand, because not possible to understand now from a naked eyes that okay it has been went and bind to it. We will use this antibody, secondary antibody, with this enzyme label antibody. And which enzyme is present with this antibody? The enzyme which has the ability to convert the substrate from one color to another. If that change of substrate happen, it means this antibody which is specific for this target antigen has went and binded to that. It means there has been the presence of that antigen. So this is how it works. So what we have studied in today's class, we basically studied about two things. First is gene therapy and other is the molecular diagnosis. So what actually is gene therapy? Gene therapy, as stated, is a collection of the processes in which what we can do, we can replace, we can functional genes into insert functional genes which has been disrupted and which has been not been working properly in the human body. So in this for today class, which example we have seen? We have seen that a 4 years old girl which is suffering from a disease skid, which is how why it is happening to her. It is happening to her because she don't have this ADA gene. There has not been presence of this ADA gene on her chromosome. So now what we did, we took the lymphocyte from that girl, okay, culture it outside. Parallelly, what we did, we took that corrected gene which is able to produce this ADA enzyme properly. Then we took a vector which is a retrovirus. Now this vector went and get into that lymphocyte, transform that lymphocyte. So now that lymphocyte can has the ability to produce what? This ADA enzyme. And now the lymphocytes which is genetically altered, which can produce this ADA enzyme, has been again reinfused into that girl's body. So once now she has this ADA enzyme, ADA gene, now she can produce this ADA enzyme. And now if she can produce this ADA enzyme, then there is no problem. But here is the thing, is that lymphocytes can't be mortal, that it lymphocytes are mortal. Okay, it means what? It will die after some time. Once that genetically altered lymphocytes will die, then there will be no production of that enzyme. So now we have to again and again insert until that lymphocyte is living, it's fine. Once it's die, we have to again inject that lymphocytes into that girl's body. So now there comes a alternative that how we can cure this permanently. How we can cure this permanently? We can take the bone marrow. We know that bone marrow has this power to produce all kind of RBCs, and now this even it can renew it. So what we can do, we can take the stem cell from bone marrow. We will insert a properly functioning ADA gene into that, and now if we will insert that into her bone marrow, now what will happen? She will lifelong can can generate this ADA enzyme. But the thing is that this has to be done in embryological state. Okay, so if this has to be done in the embryological state, it has to be detected at that stage only. That's why nowadays there are a lot of tests where we can, before this birth of the child, you can see that whether that child has genetic disorder or not. So this was all about genetic therapy, gene therapy. Then the first gene therapy was done in 1919, where we have treated a 4-year-old girl who has this ADA deficiency. Initial ADA gene therapy used engineered lymphocyte but wasn't a permanent cure. A permanent ADA cure might involve introducing the gene into the early embryonic marrow cells, as I said. Molecular diagnosis, which can be like recombinant DNA technology, PCR, ELISA, allows early disease detection. PCR has this power to detect the low pathogen or gene mutations concentration by doing what? By amplifying their nucleic acid. They can make it in the billion of copies. This molecular diagnosis also use radioactive probes. No signal indicates a mutated gene. So, as we have studied about this radioactive probe, that DNA and RNA probe. So, why it is saying that no signal indicates a mutated gene? So, for example, there is a gene. For example, there is a ADA gene, okay, and it has a particular sequence, and we have make a complementary RNA or DNA, not RNA. So we have make a complementary DNA probe, radioactive probe, and we said was like, okay, in this sample, this will go and bind to that particular gene, but it didn't got bind there. Why? Because there was some mutation, so there was no radioactive signal because they are not binding to its complementary part because there is no complementary DNA has been now present because there was some mutation. So this is saying that no signal indicates a mutated gene is there. Then ELISA detects infection by identifying what? Antigens or antibody. We have studied how we can use antibody, then there will be a target antibody, then there will be a target antigen, then there will be enzyme link antibody, then who has the ability to convert the chromogenic substrate so that we can understand or we can know that okay, there has been presence of that infection. So let's see the NCERT based questions. So first question was this: why was the initial gene therapy for ADA deficiency not a permanent cure? That why this initial gene therapy which we have used for ADA deficiency was not a permanent cure where we were using this lymphocytes. So the solution says the initial gene therapy for ADA deficiency not a permanent cure. Why? Because it involves periodically infusing genetically engineered lymphocytes. We know that lymphocytes gonna die from after a particular point. Okay, it is not immortal, it will die. So once that engineered lymphocytes will die, then there will be no production of ADA enzyme. So therefore, we need to periodically infuse that lymphocytes, genetically engineered lymphocytes. These lymphocytes are not immortal, as I said. They have a limited lifespan. Therefore, the patient required repeated infusions, making it not a permanent cure. So, what the second question says that we have to explain the principle behind ELISA for a disease detection, how we detect a disease using ELISA. So, What is the principle of ELISA? ELISA is based on the principle of antigen and antibody interaction. Infection can be detected by identifying the presence of a specific antigen. Okay. Now, these antigen can include protein, glycoprotein from the pathogen. Detecting the antibodies synthesized by the patient body against the pathogen. We have seen that how we can use this antibodies which has been synthesized by we and patient's body. So we use this antibody, then there will be the antigen, and then there will be the antibody which have this enzyme. So now we came to keynotes. So the first clinical gene therapy was done in 1990, which we have treated this 4 years old girl with ADA deficiency. Initial ADA therapy was not permanent. Why? Because the lymphocytes are not immortal, they are mortal. PCR is widely used for detecting HIV, cancer mutations, and genetic disorders. ELISA detect infection based on what? Antigen and antibody interactions. So these are the interesting questions that you guys gonna solve yourself. First question says, you guys have to define gene therapy in your own words, and how does it aim to address hereditary disease? Okay. Then you have to list 3 molecular diagnostic techniques which we have also studied. For what general purpose are these techniques particularly useful? For which purpose it is useful? We have studied that for genetic mutations or cancer, HIV, infections. You have to write it yourself. What does ELISA stand for? We studied ELISA stand for what? You guys have to write it yourself. What specific type of vector was used to introduce this ADA complementary DNA into lymphocytes? We studied which vector it is used and why it is used. So even you can write that, what its actual function is and why it has been disarmed, all those things. So thank you. So here are today's TPP, that daily practice question. So let's see first question. Which of the following best describe the primary goal of gene therapy? The question asks what is the main goal of this gene therapy. So first let's see the options. To replace all the defective gene in an individual genome. So can we replace all that defective genes in an individual's genome? Second, to introduce a functional gene to compensate for a non-functional gene causing a disease. Okay. Then third, say to mutate the existing defective gene to restore its function. Or fourth, to remove the defective gene entirely from the genome. So as you guys can see in the options, that option that you have will replace all the defective gene in individual genome. No, this is not what gene therapy used for. And we can't even mutate the existing defective gene to restore its function. It's not like that there is some mutation, we will go and cure that, correct that mutation and it will fine. No. What actually, or neither we can remove the defective gene entirely from the genome. What we do, we introduce a functional gene to compensate for a non-functional gene which is causing that disease. So here. Now what second question says? Second question says that in the first clinical gene therapy for ADA deficiency, why were lymphocytes chosen as the target cell? Why we have chosen lymphocyte as the target cells? Why not other cells? So let's see. Lymphocytes are immortal and do not require repeated infusion. Okay, so this is not something that why we have chosen this lymphocyte. This is one of the drawbacks. Second, lymphocytes are easy to culture and can be genetically modified using retroviral vectors. Yes, this is the answer, that it is easy to cultivate, culture, and it can be genetically modified using which vector? Retroviral vectors. Third option says that lymphocytes are the only cells affected by ADA deficiency. No. And the lymphocytes can differentiate into bone marrow cells. No, it is not true. So the answer is second. third question says, why is gene therapy involving early embryonic cells more effective than therapy using lymphocytes for ADA deficiency? So, what this question is saying, that why this gene therapy in which we can, treat this early embryonic cells is more effective than the therapy in which we are using lymphocyte and all those things. Let's see the options. The option says embryonic cells are immortal and do not require repeated treatments. Okay. Second, embryonic cells can differentiate into all types, ensuring permanent cure. Third, the introduced gene integrates into germline, passing to future generation. And four, the embryonic cells are more resistant to retroviral actors. So, embryonic gel is what? It is immortal and do not require the repeated treatment. That is the reason which why we are using this. And save and next. Question this says, which of the following is a limitation of enzyme replacement therapy for ADA deficiency? So it requires frequent injection and is not completely curative. Yeah. It causes mutation in the patient genome. No. It is only effective in embryo. No. It replaces the defective gene permanently. No. The answer is it requires frequent injection and is not completely curative. What role does the retroviral vector play in the gene therapy for ADA deficiency? It directly repairs the defective ADA gene in the patient cells. It delivers the functional ADA cDNA into target cells. It suppresses the expression of the defective ADA gene, or it triggers the patient immune system to produce ADA enzyme. So the question is asking us, what is the role of this retroviral vector? Okay, so what it does, it delivers the functional ADA cDNA into the target cells. So this is the all 5 questions for today's class. So now I am ending the class. Thank you.